Imagine you're a biologist, right, trekking through the Peruvian Andes. You are surrounded by this incredibly dense rainforest, and you pick up what looks like a completely ordinary, dried up, dead leaf.
Right, just a regular piece of forest floor debris.
Exactly. But then the leaf twitches.
Oh, man.
Yeah, because it's not a leaf at all. It's a toad, the Rhinella junga. And while it looks, almost indistinguishable from every other dead leaf mimicking toad in that forest, there is this one bizarre detail. It doesn't have eardrums.
Just completely missing them.
Yeah.
Which is wild, because if you aren't paying incredibly close attention to its specific anatomy, that is something you would miss entirely.
Totally. And that missing eardrum isn't just some quirky biological footnote. It's actually a clue that completely shatters how we organize life on Earth. Because for a long time, we sort of just assumed we could trust our eyes,
Yeah, which is a massive trap.
It really is. Back in 2012, researchers in Papua New Guinea were sifting through leaf litter, and they found this frog called Pedifrin emoensis, and it is barely 7.7 millimeters long.
Which makes it the smallest known vertebrate on the planet, right?
Yeah, exactly. It's practically invisible, just feeding on microscopic mites. So we have an invisible frog and then a toad that is playing like an intense game of dress-up. It makes you wonder how we ever managed to put labels on any of this stuff.
It's basically the ultimate filing nightmare. Finding a new species is an incredible rush, obviously, but it forces us to confront this really foundational problem. Like, how do we actually know what we are looking at?
Right.
If two frogs look identical, but one has no eardrums, are they the same animal? Does it even matter?
And that question right there is the mission for our deep dive today. We are exploring the surprisingly dramatic, heavily debated world of biological classification.
It gets way more dramatic than people realize.
Oh, for sure.
Yeah.
We're going to look at why the age-old method of just, eyeballing it is failing spectacularly, and how looking at the actual instruction manual of life DNA is actively rewriting the entire tree of life.
And we should clarify, the stakes here are much higher than just getting the Latin names right for a textbook. Classification, or taxonomy, is the bedrock language of biology.
Like it's the foundation for everything else.
Exactly. Without a unified system to identify organisms, a researcher in Brazil can't confidently compare notes with a researcher in Japan. You can't track how ecosystems are collapsing or thriving. You can't identify new plant compounds for life-saving medicines.
Wow.
And you definitely can't protect an endangered species if you can't definitively prove it exists in the first place.
Which brings us back to the most basic tool humans ever had, right? Our eyes. Before we had microscopes or gene sequencers, we just looked at things and grouped them by their physical appearance.
Yeah, the old, if it looks like a duck method.
Right. If it has fur and claws, it goes in this box. If it has green leaves, it goes in that box.
And that is the human brain's default setting, honestly. We categorize visually. So in the 18th century, a Swedish botanist named Carolus Linnaeus decided to formalize that impulse.
He's the guy who built the morphological species concept, right?
He is. And morphology simply refers to the study of form or shape.
Okay, let's unpack this. So he was judging books entirely by their covers, or I guess by their pages if he was looking at inner anatomy, but still purely physical traits.
That's the idea, yeah. Linnaeus categorized species almost entirely on their physical structure. He's the architect of the binomial system.
The two-part Latin name thing?
Exactly. The universal identifier given to every species. The first name is the genus, which groups closely related cousins together, and the second is the specific species.
So that's where we get Homo sapiens for humans, or Vanessa atalanta for the red admiral butterfly.
You got it. And Linnaeus didn't just stop at two names. He built a whole filing cabinet, this massive hierarchy, going from the broadest category down to the specific organism.
Right, domain, kingdom, phylum, class, order, family, genus, species. And our sources actually shared a great mnemonic for keeping that straight, which I love.
Oh, what is it?
Desperate King Philip came over for great spaghetti.
That is fantastic. I haven't heard that one in years.
It's so catchy. But, okay, despite how clean and structured that system is, the problem is that relying purely on that morphological approach is a massive trap. Nature is incredibly deceptive.
It really is.
I was trying to visualize the flaw here, and organizing species by how they look seems a bit like trying to organize a massive city library by the color of the book covers instead of the plot.
Oh, that's a perfect way to look at it.
Right, because you end up with a huge shelf full of blue books, but one is a French cookbook, one is a calculus textbook, and one is a fantasy novel. They have absolutely nothing to do with each other, but your visual system says they belong together.
And the biological equivalent of that blue bookshelf is a phenomenon called convergent evolution. This is what happens when two completely unrelated organisms face the exact same brutal environmental challenges.
And they independently invent the same physical solutions.
Precisely. The sources bring up a really great example with desert plants, agaves and aloes.
Oh yeah, if you put an agave plant from Mexico next to an aloe plant from South Africa, they look like identical twins.
They really do. Both have thick, fleshy, spiky leaves arranged in a rosette, to conserve water in the scorching heat. You would bet your life savings they share a recent common ancestor.
But they don't at all.
Nope. They evolved those traits completely independently on opposite sides of the planet. Biologists call these physical coincidences analogous features.
Analogous features, okay?
Yeah, think of the torpedo-shaped bodies of a shark and a dolphin. One is a fish, one is a mammal. They look similar because water hydrodynamics demand that shape, not because they're closely related.
Wow. So if a visual system is constantly being tricked by these analogous features, what is the alternative? How do scientists cut through the disguise?
Well, they have to hunt for homologous structures.
Yeah.
These are features that genuinely prove a shared biological origin, like a true family trait, even if the outer packaging has changed over millions of years.
But spotting those with the naked eye is incredibly difficult.
It is. Plus, morphology has an even bigger vulnerability, which is sexual dimorphism.
Ah, the peacock problem.
Exactly.
Because if I were an alien arriving on Earth with Linnaeus's rulebook, and I saw a male peacock with a 6-foot-long iridescent train of feathers, and then a female peahen, which is essentially just a brown camouflaged bird.
You'd totally separate them.
Yeah, there is no way I would put them in the same species box. Visually, they are completely distinct.
The morphological model breaks down entirely there. So biologists knew they needed a more robust definition, something that wasn't reliant on subjective visual comparisons. That's when the scientific community shifted toward the reproductive species concept.
Right, and the rule for this one seems pretty straightforward at first glance. A species is a group of organisms that can interbreed to produce fertile offspring.
Yeah, it's not about what you look like. It's about whether you can successfully mix your genes. It focuses entirely on gene flow. If genetic material can travel between individuals and result in a viable, fertile new generation, they are part of the same genetic family.
I remember learning the classic example of this, the horse and the donkey.
Oh, yes, the classic.
They are clearly similar, they can mate, and they produce a mule, but that mule is sterile. It cannot pass those genes forward. The flow stops dead. Therefore, horses and donkeys are definitively separate species. It feels completely airtight.
It does feel airtight
Yeah.
until you realize that nature refuses to be boxed in by human definitions. Think about lions and tigers.
Okay.
They are undeniably different species. They occupy different habitats, have totally different social structures, and look nothing alike. But if a lion and a tiger mate in captivity, do you know what happens?
Well, our sources point out the loophole here. The offspring, the ligers or tigons, are actually frequently fertile.
Yes, they can reproduce. So, under the strict rule of the reproductive concept, the boundary between lion and tiger suddenly dissolves. And this isn't an isolated incident.
Really?
Yet many plant species frequently cross-pollinate with distant relatives to produce perfectly fertile hybrids. The boundaries are way more porous than we thought.
And, it completely ignores organisms that don't need a partner at all. How do you apply a breeding rule to a single-celled bacteria that reproduces by just cloning itself?
You can't.
Or to a Tyrannosaurus rex, where all we have are bones and literally zero data on its mating habits?
Exactly. The visual rule fails because of mimicry, and the reproductive rule fails because of hybrids and asexual organisms. Taxonomists hit a brick wall. They realized that looking at the outward result, the animal itself or its offspring, just wasn't enough. They had to read the underlying blueprint.
Which brings us to the molecular revolution. Here's where it gets really interesting, because we start measuring jaw bones and feathers, and we started looking at the invisible architecture, right? DNA, RNA, and proteins.
This is the era of molecular phylogeny. And the premise is breathtakingly simple, even if the actual science is complex. The closer the evolutionary relationship between two organisms, the more similar their molecular code will be.
Right.
To read that code, scientists developed techniques like gel electrophoresis.
The sources mention that process, but I want to make sure I really understand how we can literally see differences in molecules. How does the gel actually do the sorting?
Okay, picture a massive, incredibly dense forest.
Okay, dense forest. Got it.
Now imagine a huge crowd of people of all different sizes trying to run through that forest at the same time because they're being pulled by this powerful magnetic force.
Oh, I see. So the dense forest is the gel, and the magnetic force is an electrical current.
Yes, exactly. Scientists take a sample of DNA fragments and place it at one end of the gel, then turn on an electrical current. DNA has a negative charge, so it naturally gets pulled through the gel toward the positive end.
And I'm guessing the smaller people in your analogy, like the smaller DNA fragments, can slip through the dense trees of the gel much faster than the larger ones.
You nailed it. The fragments separate based on their size and electrical charge. When the current is turned off, you are left with this distinct series of bands. It looks like a unique molecular barcode for that specific organism.
Oh, wow. So when you line up the barcodes of a dozen different frogs, you can instantly see who is related to who. You bypass the whole eardrum issue entirely.
Completely.
That makes perfect sense. And the sources highlight a story where this wasn't just some abstract laboratory exercise, it actually saved global agriculture. Let's talk about the Fusarium graminearum fungus.
This is a perfect example of molecular taxonomy in action. So for decades, farmers and plant pathologists worldwide were fighting this devastating disease called head blight or scab, which attacks wheat and barley. It was an absolute agricultural nightmare, literally threatening the global food supply.
And everyone just assumed they were fighting a single enemy.
Because morphologically, it was a single enemy. Whether you looked at a sample from a wheat field in North Dakota or a barley farm in Europe, the fungus looked identical under a microscope. It caused the exact same rotting symptoms in the plants.
So the scientific consensus was unified. One species, Fusarium graminearum.
Right. But then geneticists started running the DNA and protein profiles of these fungal samples from around the world.
Yeah.
And the barcodes didn't match.
The visual similarity was a complete illusion.
Total illusion. The genetic profiling revealed that this one pathogen was actually at least 8 genetically distinct species hiding behind the exact same morphological mask.
That is insane. Imagine spending millions of dollars trying to formulate a specific fungicide, believing you were targeting a single biological mechanism, only to find out you were fighting 8 completely different biochemical enemies.
It revolutionized how agricultural science treats the disease. It proved that without molecular data, we are often flying blind. But, this shift to genetics introduced a brand new problem. Sheer volume.
Right, because sequencing genomes isn't like looking at a handful of physical traits.
Exactly. You are looking at millions, sometimes billions, of base pairs of DNA.
You can't just put that on a chalkboard. A human researcher would spend a lifetime just trying to find the patterns in a single strand of wheat DNA.
Which birthed an entirely new discipline called bioinformatics.
Yeah.
We basically had to build massive computational networks and specialized algorithms just to store, organize, and analyze this ocean of biological data.
I try to picture bioinformatics like a massive, hyper-advanced text search engine. If you want to prove 2 animals are related, you can't just look at the whole book.
Right.
The software aligns millions of letters of DNA code side by side, rapidly searching for exact matching sequences. It's like trying to find one specific sentence that appears in a million different books simultaneously.
That computation is what allows us to trace evolutionary lineages back billions of years. And when scientists unleashed bioinformatics on the very foundations of biology, the results were incredibly disruptive.
You're referring to the textbook rewrite. Because I definitely grew up learning the 5-kingdom model.
Oh, me too.
Yeah. All life was sorted into animals, plants, fungi, protists, which was kind of the junk drawer for algae and weird single-celled things, and Monera, which was the kingdom for all bacteria. It was a very comforting, tidy way to look at the world.
It was tidy, and it was entirely wrong.
Right.
In the late 1970s and 1990s, a microbiologist named Carl Woese began using molecular phylogeny to examine ribosomal RNA. Ribosomes are the tiny factories inside cells that build proteins. Because every living thing needs them to survive, they evolve extremely slowly.
Making them the perfect molecular clock to look deep into the past.
Exactly. And when he used that text search engine on the bacteria kingdom, Monera, what he found was a monumental error.
Okay, what was the error?
The organisms we had comfortably grouped together as bacteria were actually 2 completely different forms of life. Genetically speaking, the differences between these 2 groups of microscopic blobs were as vast as the difference between a human being and a mushroom.
That is staggering. It forced the creation of a whole new taxonomic level above kingdoms, right?
Yes, the 3-domain system. We now classify all life into bacteria, Eukaryota, which contains us, the plants, the animals, and fungi, and this newly discovered third domain, Archaea.
Okay, the Archaea are the ultimate biological curveball. Initially, researchers called them archaebacteria and assumed they were just some ancient, hardcore offshoot of bacteria that lived in insane environments.
Right, the extremophiles. Boiling hydrothermal vents at the bottom of the ocean, highly acidic hot springs, subglacial lakes.
But as our genetic tools became more sensitive, we realized Archaea aren't just clinging to the edges of volcanoes, they are ubiquitous. They are in the ocean, in the soil, in your backyard, everywhere.
But they are emphatically not bacteria.
Not even close.
In fact, the genetic data reveals something that sounds almost impossible. The Eukaryota domain, again, that's you, me, trees, and whales, shares a more recent common ancestor with the extremophile Archaea than we do with the standard bacteria living inside our own stomachs.
That fundamentally alters how I view my own biology. And it actually sets the stage for what I think is the most mind-bending concept in the entire source material.
The endosymbiotic theory.
Yes. This theory explains the leap from those simple, single-celled organisms to the complex life we see today. Because eukaryotic cells, the building blocks of the human body, are incredibly complex pieces of machinery.
They really are. We have a distinct nucleus that vaults our DNA, and we have mitochondria, which are specialized organelles that generate all our cellular energy. Bacteria and Archaea don't have any of that internal machinery.
So the obvious question is, where did our cells get these specialized engines?
And what's fascinating here is, the theory suggests it wasn't a gradual mutation, but a microscopic heist. Billions of years ago, a large ancestral cell encountered a much smaller free-living bacteria that was really good at using oxygen to make energy. And the large cell literally swallowed it.
But crucially, it didn't digest it.
Right. It was an integration.
The larger cell provided a protected environment and a steady stream of nutrients, and in return, the engulfed bacteria pumped out massive amounts of efficient energy.
It is the ultimate biological merger. Over millions of years, that swallowed bacteria lost its ability to live independently and became a permanent integrated organelle, the mitochondria.
Wow.
And this same process happened later with early plant cells, which swallowed a photosynthetic bacteria that eventually evolved into chloroplasts, giving plants the ability to turn sunlight into food.
That honestly sounds like a crazy science fiction story. Is there actual molecular proof that our energy factories used to be free-floating bacteria?
There is a total smoking gun. If you look inside a human cell, the nucleus holds your human DNA. But if you look inside the mitochondria, they have their own separate circular DNA.
Wait, really?
Yes, and that DNA looks remarkably similar to modern bacterial DNA. It is a leftover genetic fingerprint from an ancient cellular collision.
DNA just refuses to keep nature's secrets. It exposes everything. And it's not just happening at the microscopic cellular level. Biochemical analysis is connecting massive, familiar animals in ways that completely defy logic.
It really exposes the limitations of those old visual models we talked about. Let's do a quick thought experiment. Picture a starfish,
Okay.
a 5-armed marine invertebrate covered in a tough skin, walking along the seafloor on hundreds of tiny tube feet.
Okay, got the starfish pictured.
Now, what land animal would you assume is its closest evolutionary relative?
Based purely on vibes and morphology, I would guess something like an insect, a crab, or maybe an earthworm, something without a backbone.
That is exactly what early taxonomists thought.
Oh?
But if you look past the exoskeleton and examine their biochemistry, specifically how their muscles store and synthesize energy, you get a massive shock.
Let me guess. They aren't related to worms at all.
Not at all. There is a specific molecule called phosphocreatine. It is a critical component in synthesizing ATP, which is the chemical energy currency used in muscle contraction.
Okay.
Phosphocreatine is found almost exclusively in the muscle tissue of vertebrates. Humans use it, dogs use it, sheep use it.
And what do invertebrates like worms and insects use?
They use a totally different molecule called phosphororigine. But when biochemists analyzed the muscles of echinoderms, the group containing starfish and sea urchins, they didn't find phosphororigine. They found phosphocreatine.
So biochemically, the starfish is operating its muscles like a sheep, not like a worm.
Exactly. This molecular breadcrumb, along with data from how their embryos develop, proves that starfish and vertebrates share a common evolutionary ancestor deep in the past, completely separate from the lineage of insects and worms.
You could stare at a starfish and a sheep for 1,000 years and never, ever make that connection without molecular data.
Never.
But I want to pivot here, because knowing a starfish is related to a sheep is fascinating, but how does this new molecular rulebook actually help us today? Does taxonomy actually matter for the animals currently struggling to survive on this planet?
Oh, it matters immensely. Molecular taxonomy isn't just an academic history lesson, it is a vital tool for conservation. It can actually help us correct the devastating mistakes of the past. Let me tell you the story of the quagga.
Okay, I've seen historical illustrations of this animal, and it looks like a creature from a myth, like half horse, half zebra.
The quagga was a magnificent animal native to the plains of South Africa. Visually, it was stunning. The front half of its body, the head, neck, and shoulders, was covered in bold, zebra-like black and white stripes.
Right.
But as you moved down its back, the stripes faded away into a solid chestnut-brown coat, ending in white legs and a white tail.
Looking at that under Linnaeus's morphological rules, it's a slam dunk. That is obviously a unique, distinct species.
Which is exactly how it was classified, Equus quagga. Unfortunately, European settlers hunted the animal ruthlessly for its meat and its unique hide. The populations collapsed, and the very last known quagga died in captivity at the Amsterdam Zoo in 1883.
That is so tragic.
It really is. For over a century, the scientific community mourned it as a completely extinct, irrecoverable species.
Until the molecular revolution arrived.
Precisely. In the 1980s, the field of ancient DNA started to mature. Researchers realized they didn't need living tissue to extract genetic data. They took museum taxidermy mounts of the quagga specimens that had been stuffed and sitting in glass cases for over 100 years.
And they managed to extract mitochondrial DNA from fragments of dried skin and muscle.
They did.
That is basically biological necromancy. What did the text search engine find when they sequenced it?
A massive taxonomic plot twist. When they compared the quagga's DNA barcode to modern equines, they realized it wasn't a distinct species at all. Genetically, the quagga was nearly identical to the plain zebra, which is still abundant in Africa today.
Wow.
The differences were so minor that the quagga had to be reclassified as a mere subspecies, basically a regional color variant. Its new name became Equus quagga quagga.
So the lack of stripes wasn't a deep evolutionary chasm, it was just a recent cosmetic adaptation. I think the sources mentioned that the brown coat might have provided better camouflage on the dry, dusty plains, or maybe it had to do with insects.
Yes. Stripes are actually a known evolutionary defense mechanism against biting tsetse flies. The quagga lived in a specific region of South Africa that was largely free of tsetse flies, so the evolutionary pressure to maintain the full-body stripes relaxed, allowing the brown coat variation to dominate.
But the real revelation here is the genetics. Because if the quagga is just a cosmetic variation of the modern plains zebra, that means the genetic recipe, the DNA instructions that dictate that half-striped brown coat, didn't actually vanish in 1883.
It was never extinct. The specific alleles or gene variants responsible for the quagga's appearance were just diluted, floating around quietly in the massive, living gene pool of the modern plains zebra.
And this realization sparked a massive conservation initiative, right? The South African Quagga Project. But how do you actually bring an animal back if you aren't cloning it?
You use the power of selective breeding, heavily informed by taxonomy. The researchers went out and examined thousands of living plains zebras. They looked for individuals that naturally exhibited a slight reduction in striping on their hindquarters, or maybe a slightly browner tint to their coat.
Oh, I see. They found the zebras carrying the dormant quagga traits.
Exactly. They grouped these specific zebras together in a protected reserve and let them breed. When the foals were born, they selected the ones that looked most like the historical quagga and bred them with other heavily selected individuals.
So generation after generation, they are systematically concentrating those specific genetic alleles back into a single population.
It's essentially reverse engineering the extinction, and it's working. They have successfully produced a population of animals they call rau quaggas, which look incredibly similar to the 19th century paintings of the original animal. They are currently roaming the Western Cape of South Africa.
That is a profound success story.
It really is.
Yeah.
And it perfectly encapsulates why this transition in how we classify life is so critical. We have moved from the flawed naked eye of the 18th century to the molecular microscopes of the 20th century, and now to the staggering computational algorithms of bioinformatics. We aren't just memorizing Latin names anymore.
Right. We are decoding the ancient, invisible blueprints of life, revealing connections that completely redefine our place in the natural world.
Precisely.
It proves that the natural world is so much more fluid and interconnected than we ever gave it credit for. Which leaves you with one final, fascinating thought to ponder. If the extinct quagga was never really gone, if it was just hiding quietly inside the DNA of the modern zebra, waiting for scientists to coax it back out into the sun, what other extinct traits or ghosts of ancient creatures might currently be walking the earth right now, locked silently away in the DNA of the animals sitting in our own backyards?